The promoter DNA sequences is where RNA polymerase binds to begin transcription, but repressors can block transcription. So, when we think about eukaryotes, we think about enhancers, promoters, and genes. And it's at the enhancer where we have these activators binding, and at the promoter region we have transcription factors and other mediator proteins that bind to that promoter so that RNA polymerase can hook up, and it's as that activator puts all this stuff together that transcription can actually begin in eukaryotes. Now, there are regulatory sequences that make repressors, and repressors can interact in eukaryotes and prokaryotes. So when we're seeing prokaryote genes, we are going to often see operators, and that's where the repressor will bind. In eukaryotes, repressors can bind in different locations to interrupt the process of transcription. But as we look here, again, RNA polymerase and this regulatory gene, there is no operon, so the RNA polymerase can bind and it can begin transcription right away. And as we're making messenger RNA, that messenger RNA will find a ribosome and it will begin translation. And we'll start gathering these amino acids together with transfer RNA, and we have our primary structure, our secondary structure, which becomes a tertiary structure, a 3D dimensional activated protein, and that's where our repressor comes in. So we looked at this inducible operon where the repressor sits on the operator and RNA polymerase cannot bind. So in inducible operons, the gene, the functional structural genes are off unless an inducer removes the repressor. So when we look at these other repressible operons like the trip operon, these are on all the time because these structural genes assist in making proteins for absolutely necessary life processes. So in humans, we get tryptophan by eating other organisms, and by eating those other organisms that have tryptophan in them, we get tryptophan as a raw material within our body. However, we cannot create tryptophan on our own, but bacteria and plants can. So they don't have this amino acid naturally because they're not eating it. They have to create it through gene expression. So when we're looking at this, tryptophan is not, it's absolutely essential, but it's not there all the time. They have this precursor molecule, but it's only through the transcription and translation of these particular genes that can transform the precursor molecule into tryptophan. So as we start here, again, there's no operon or operator sequence down here. So RNA polymerase can bind to the promoter, which initially begins transcription. And at transcription, we make messenger RNA. It goes to the ribosome. Transfer RNA brings over amino acids. We have our primary structure. We end up with our secondary structure, and we end up with our tertiary structure. Now, this protein that's created, this regulatory protein that's a repressor, in these operons, it's inactive. So although we have a repressor made, and we can make unlimited amounts of repressors, they are made in an inactive state. So when we go up to this one, again, this operon is in an on position because this is inactive and it's not shutting down the operator. So the RNA polymerase binds the promoter and it progresses right past the operator to the first gene and begins transcription. Our messenger RNA is read at the ribosome. We go through translation, join our amino acids into our primary structure. We get our secondary structure and our tertiary structure. And it's this functional protein that can act as an enzyme to convert this precursor molecule into the next sequence. Now, this still isn't tryptophan. We have to use these multiple structural genes to get it to that point. So at this point, this next gene is being read. We get our next transcript, and we go through translation. We get our primary structure, our secondary structure, and our tertiary structure of our protein. And this enzyme can now take the next step. And we have this metabolic pathway where these enzymes are converting an initial substance into a secondary substance, and on and on and on until we get to where we want to go. So again, RNA polymerase is reading gene after gene after gene because these structural genes are all regulated together. So RNA polymerase makes the next transcript, which goes through translation, and we get our protein enzyme, which then ultimately gives us tryptophan. And yay, we have tryptophan. But at this point, this process would continue because, again, there's no repressor on the operator. So RNA polymerase would head towards a promoter. But in this case, that we just went through a lot of work to create tryptophan, and we have tryptophan present. So an efficient way to do this process is to now turn this off. It takes a lot of work to go make tryptophan. If we have tryptophan present, we don't need to make more tryptophan. So the tryptophan turns our inactive repressor into an active repressor, which can now bind to the operator sequence and turn these genes off. RNA polymerase cannot progress, and because we have tryptophan, we don't need to make the enzymes to make more tryptophan, and it is being repressed. So again, we have these regulatory gene sequences that make repressors, and repressors bind to operators in prokaryotes. That turns genes off, and the structural genes downstream would not be able to be read and transcribed. When we look at this process, again, we make our repressor, but it's in an inactive state. So at this point, this cannot bind to the operator sequence, and the RNA polymerase will hook onto the promoter, and you'll get all of those enzymes being made through transcription and then translation. And it's all of these enzymes that end up making that tryptophan molecule. And it's the tryptophan molecule that ultimately binds with the repressor to make it active and turn off the genes to make more tryptophan. By this active repressor binding to the operator sequence, RNA polymerase cannot proceed with transcription. So promoters are where RNA polymerase binds to do transcription, but repressors block transcription.